There are now probably hundreds of labs around the world that are actively trying to understand the language of the human embryonic stem cell. In other words, how does that cell first know to keep dividing, maintain its potential, and then how does it know to become a particular kind of mature, differentiated cell? Now, for a number of years, many of us in the field have understood or tried to understand the genes that are involved, the genes that signal that a stem cell should become this cell type or that cell type. But what we know is that genes really are not the workhorses of a cell or of the body. Genes simply encode or kind of lay down the blueprint for proteins. It's proteins that really actually do the work. And studying that area is called proteomics.
Very briefly, protein phosphorylation is like an on-off switch for protein activity. As Dr. Snyder mentioned, the proteins are really the workhorses in the cells in controlling what the cells do. So phosphorylation is like an on-off switch, whether they should continue to self-renew, which is a pluripotent state he mentioned, whether they should differentiate, if so, how they should differentiate, if it turns out they should die, if not, just any cell behavior really. This is termed cell signaling. You can think of those phosphorylation sites as specific players in telling the cells if they should become neurons, if they should become liver cells, if they should become skin cells, etc. And it's a very complex mixture of phosphorylation sites that actually dictate what the cells will do in this cell signaling that I mentioned.
I think that this kind of study, and particularly the laundry list of proteins that are unveiled, give us really intriguing drug targets. Now, the drug targets can be thought of in two ways. One, maybe these are ways to take these cells and turn them in a dish into the kind of cells that you would want to transplant. But interestingly, another way of looking at it too is revealing certain pathways that might be important in the body itself, so that one can now tailor drugs to take a person's own stem cells and either direct them in useful ways, or the flip side of that is to prevent them from becoming undesirable cell types, for example, becoming cancers. So I think that once one understands the fundamental language of the stem cell, and that's what essentially proteomics really is, we have unlimited possibilities of now using this knowledge for health and repair.